The title of this PowerPoint is Cell Reproduction. And certainly when we regulate the number of cells in an organism, one of the primary factors there is the rate at which cells reproduce. But another factor is the rate at which cells die. And so at the end of this PowerPoint, we're going to be talking about the various processes that are important in cell death. We've already spoken about one process of cellular reproduction, and that is meiosis. Meiosis is the process of cell reproduction when we make gametes or sex cells. Remember that we have spermatogonia or oogonia in males and females respectively as the precursors to the sperm and the egg. Those spermatogonia and oogonia are called because those cells have both a mom copy and a dad copy of each of the chromosomes. The DNA is copied, and that process is termed DNA replication. So that in that middle cell, you see that there are two dad copies of the big blue chromosome, two dad copies of the little blue chromosome, and two copies of the mom big chromosome and two copies of the mom little chromosome. During the cellular division, the first cellular division of meiosis, those chromosomes are distributed such that each of the daughter cells get either a mom copy or a dad copy of each of the chromosomes. So those cells are termed haploid. Diploid is when you have both a mom copy and a dad copy of each chromosome. Haploid is where you have either a mom copy or a dad copy of each of the chromosomes. The cells then will undergo a second division. The haploid cells will undergo a second division without copying the DNA, without DNA replication, and will undergo a cell division in which each of the daughter cells that are produced are not identical to one another. So they have, in the case of that last row, non-identical haploid gametes at the end of 2 rounds of meiotic division. Mitosis is the process of cell reproduction in all of our other cells, in the non-gametes, in the non-sex cells. We start out with an original cell that undergoes DNA replication, so each of the chromosomes, mom and dad, are copied. Those chromosomes are distributed such that the daughter cells get both a mom copy and a dad copy of each chromosome. This produces two daughter cells that are identical to one another. Sometimes we refer to these as clones. Mitosis, that process of cell division, is only a small part of what we call the cell division cycle. Here is mitosis, only a wedge of this pie, where you can see the majority of time in the cell division cycle is spent in something called interphase. Interphase is all of this stuff, all of the time spent non-reproducing. And this interphase is divisible into 3 separate phases. We have a G1 phase of interphase during which growth occurs, and the cells that are hanging out in the G1 phase of interphase are sort of gathering information from the environment to determine whether or not the environment would support more cells, so would support cellular reproduction. If the environment will support, there's enough room, there's enough food, etc., then the cell will enter the S phase of interphase. S phase is where the DNA is replicated. Once the DNA is replicated, the final phase of interphase is known as G2. Of course, growth is going to continue, but the cell is also going to be surveying not only the exterior environment, but also the internal environment to determine whether or not the DNA was replicated properly, whether or not there are sufficient mitochondria to be distributed into daughter cells, etc. So the assessment that's going on during the G2 phase of interphase is more or less an assessment of the internal environment of the cell. I want to look at the steps of cell division, and then I want to look at specifically the steps of the mitosis part of the cell division cycle. So first off, on the left, we have a cell, and that cell will enter the G1 phase of interphase, meaning that it will grow. If the environment is appropriate for cell division, the DNA will be replicated during S phase. The cell will then enter G2 phase of interphase to determine whether or not the internal environment in the cell is okay for the cell to enter mitosis. Mitosis allows for the distribution of the mom's chromosomes and the dad chromosomes such that each of the daughter cells will receive one of each pair of chromosomes. The cell will then divide into two daughter cells. A contractile ring will form between the two sets of chromosomes. That contractile ring will pinch the cells together, or pinch that middle part together such that two daughter cells will be created. There are multiple steps in the process of mitosis. Of course, on the left-hand side, we have a cell that is in interphase. And when we look at the cell, which is in that sort of top series of panels, the bottom series of panels is showing you sort of a cartoon version of an actual microscopic image of a cell. In that interphase cell, we notice that we don't see distinct chromosomes in the nucleus. However, after the DNA has been replicated and the cell has determined that the interior environment is suitable for entering mitosis, or the actual process of cell division, the chromosomes start to condense. So you can see in the image, the second image there, that the DNA is appearing darker. That means that the DNA is actually tightening into what are visualized as X-shaped chromosomes. The other thing that happens during this first phase of mitosis is we lose the nuclear membrane. The nuclear membrane starts to dissolve. This first phase of mitosis is known as prophase, and in prophase, the chromosomes do condense, but spindle fibers also start to grow out of structures inside of the cell known as centrioles. These spindle fibers will be important in the distribution of chromosomes into each of the daughter cells. So during prophase of mitosis, DNA will condense, the nuclear membrane will disappear, and spindle fibers will grow. The next step of mitosis is metaphase. In metaphase, the chromosomes, the condensed chromosomes, align along a midplate or equator of the cell. You can see that they are distributed such that one of the arms of each chromosome is facing one direction toward the top of the cell, and the other arm is facing toward the bottom of the cell, in the cartoon image anyway. The next phase of mitosis is anaphase. So during metaphase, the chromosomes line up along the equator of the cell. During anaphase, those lined up chromosomes separate so that you see in the cartoon image that one arm of each of the mom chromosomes and each of the dad chromosomes will move toward the top of the cell, being pulled that direction by the spindle fibers. And one arm of each of the chromosomes is being pulled toward the bottom, again by the spindle fibers. Anaphase is where the chromosomes are separated into what will be the future daughter cells. During telophase, which is the last phase of mitosis, that contractile ring forms pinching between two of the daughter cells. The other thing that occurs is that nuclear membranes appear so that the nuclei are becoming separated. Finally, following telophase, we have the process of cytokinesis. Cytokinesis is where the two daughter cells are ultimately separated from one another. So at the top of this slide, you can see a summary of each of the phases of mitosis. Prophase, where the copied chromosomes are packaged, the nuclear envelope dissolves, and the spindle fibers form. Metaphase, where the chromosomes line up in the middle so that they can be properly distributed. Anaphase, where the chromosomes separate to opposite sides of the cell. And telophase, where the nuclear membranes reform and a contractile ring causes the initial steps of what eventually will be separation of the two daughter cells. This image just shows you those latter steps, telophase and cytokinesis. Again, during telophase, this ring of microfilaments, actin and myosin, will contract to pinch the middle section of what eventually will be the two daughter cells. You can see in the middle image the contraction of actin and myosin pinches tighter, eventually squeezing tight enough so that the two daughter cells can separate. That separation of two daughter cells, again, is termed cytokinesis. The cell cycle starts over following cytokinesis. That's why it's called a cycle, because it occurs over and over and over again, unless, of course, it doesn't. There is a reason for cells to enter something referred to as a G0 rest phase between the G1 growth phase and the S DNA replication phase of interphase. In other words, there is an opportunity to get off the treadmill, to leave the cell cycle, if a cell does not want to undergo cell division again. Why would that occur? If the environment is unfavorable, or if the DNA is damaged, a cell would not want to copy damaged DNA, or a cell would not to reproduce if the environment would not support an additional cell. In other words, if there's not enough food, if there's not enough room, something like that, a cell would enter a G0 rest phase before entering the cell division cycle again. Just because you leave the cell division cycle does not necessarily mean you can't go back. If, for example, we're talking about skin cells, we don't necessarily need to make more skin cells unless we have cut ourselves, and so we need to fill in a wound. Well, that creates room for additional cells. So those cells would undergo, your skin cells would undergo cell division until the wound was healed. Once the wound was healed, the cells would realize that there is no more room for additional cells, so it would stop growing. So this is a summary slide distinguishing between the two types of cell division that we've spoken about in this class, mitosis versus meiosis. In mitosis, cells divide once after replicating the DNA. Daughter cells receive one chromosome from mom and one chromosome from dad. Daughter cells are therefore diploid, and daughter cells are identical to one another. During the process of meiosis, cells divide twice, once after DNA replication and a second time without DNA replication. Daughter cells of the first division, that first round of division, receive either a chromosome from mom or a chromosome from dad. Daughter cells of the first division are haploid, and they remain haploid throughout that second division. At the end of the second division, daughter cells are not identical to one another. In addition to cell reproduction, we also have to consider the rate at which cells die when we are regulating the number of cells in a multicellular organism like a human. There are two different ways that cells can die. The first of those is by a process called necrosis. You can see in the necrotic cell on the left-hand side that this is a very messy process. A cell essentially bursts, spewing its contents into the extracellular space. Some of those cellular contents might be, for example, digestive enzymes from the lysosomes. So releasing digestive enzymes into the extracellular space is not necessarily a good thing for the neighboring cells. A second type of cell death is referred to as apoptosis. Apoptosis is programmed cell death or controlled cell death. And looking at the image on the right, you can see that apoptosis is a fairly clean process. Cells sort of digest themselves from within and ultimately signal the immune system to clean up the debris. Apoptosis is a normal process. This image shows a couple of examples in the animal kingdom where apoptosis occurs. For example, when a tadpole's tail gets reabsorbed, that occurs via that programmed cell death. Similarly, each of us during development have webs between our fingers. If development occurs properly, those webs disappear by the time we're born. The process of apoptosis drives the dissolution of those webs between our fingers. Apoptosis is very critical in the development of the central nervous system. Remember, we were talking about later on in nervous system development when the nerves sort of remodel, the brain sort of remodels itself such that certain nerves are retained and other nerves go away. The nerves that go away are those nerves that are not used as frequently or cannot make proper connections with other neurons or other downstream cells. The process by which those nerve cells go away is apoptosis. They undergo this programmed cell death, and they do that because they're not receiving the proper survival factor signals. Apoptosis is a clean death because, as I said, cells are digesting themselves essentially from within. You can see in the little cartoon image at the top that if a cell is undergoing apoptosis, the first thing that happens is the cell begins to shrink and the DNA, labeled here chromatin, starts condensing. The DNA will ultimately start breaking apart, fragmenting it into little tiny pieces. Proteins inside of the cell are degrading, and that accounts for cell shrinkage. Mitochondria start to fall apart, releasing their contents, and that in fact drives a lot of the apoptotic process. Ultimately, flags or tags are placed on the cell surface of a cell undergoing apoptosis, such that the phagocytic immune cells, neutrophils and macrophages, can engulf the apoptotic bodies or the fragments of a cell that are ultimately produced. There are two reasons that a cell might undergo apoptosis. The first of those is what's called the intrinsic pathway. The intrinsic apoptotic pathway occurs when a problem is sensed from inside of the cell. For example, if there's DNA damage, if there's not enough oxygen, if there are too many cells, a cell will undergo apoptosis via an essential cellular suicide. There's a lot of information on the image, and I don't care that you know that. I just would like you to recognize that if the internal environment is sensed to be inappropriate, a cell in a multicellular organism can undergo apoptosis if it believes that killing itself is for the greater good of the organism. In other words, our cells will take one for the team by killing themselves via this apoptotic process. A second reason that a cell might undergo apoptosis occurs via an extrinsic pathway. The extrinsic pathway occurs if a cell is virally infected or if it's not receiving the proper communication. In this case, or in fact in the former case, if a cell is virally infected, a killer lymphocyte or some other immune cell will attach to the infected cell and essentially signal the cell that it should kill itself, undergo apoptosis, and in fact the killer lymphocyte will very strongly encourage the infected cell to undergo apoptosis, this clean program cell death.